Gels are used in molecular biology to separate and purify nucleic acid fragments, usually DNA, of different sizes. A dye is added to be able to see the fragments under ultraviolet light. The samples are loaded into the wells of the gel and separated by size using an electric current applied across the gel in a running buffer. DNA is negatively charged and it will make its way towards the positive electrode. The smaller the DNA fragments are, the easier it will be for them to worm their way through the gel. Longer fragments will find this more difficult and will take much longer to make their way through the pores of the gel. The size range of DNA fragments that can be separated by agarose in this way is around 100 to 20,000 base pairs. Agarose gels are made with varying concentrations of agarose, depending on the size of the DNA fragments you want to separate or purify. Usually this is between 0.4% and 3%. If you want to separate or resolve large fragments, then you will need a lower concentration of agarose, so that the fragments can make their way through the pores in the gel more easily. So it follows that if you want to separate small fragments, then you will need a high concentration of agarose. To make an agarose gel, you will need agarose powder and a running buffer that you dissolve the powder into. For a 1% agarose gel, you would need 1 gram of powder in 100 ml of buffer. This will vary depending on the size of fragments you want to resolve. The buffer is usually a tris borate EDTA buffer, shortened to TBE, or tris acetate EDTA, shortened to TAE. These two components are heated together to make the gel. Firstly, make up the 1 times buffer you need using the concentrated stock. We are using 5 times buffer, so need to add 400 ml of water to 100 ml of concentrate. After deciding the percentage gel you require, then you need to weigh out the precise amount of gel powder you need, in a volume suitable for the container or gel tray you're going to use. The volume usually used is around 50 to 100 ml for smaller gels. Add the required volume of buffer to a suitable container, one which will allow the liquid to expand when it's boiled and not to overflow. Carefully add the agarose and ensure that there are no lumps of powder before heating in the microwave. Place a lid or sponge bung loosely on the top. Do not tighten the lid. This is very important as the liquid can become superheated. The simplest way to heat in the microwave is in intervals of half a minute or so. Using safety gloves and goggles, carefully swirl the mixture in between intervals to ensure that there are no particles of powder left. These appear as tiny specks, leaving a trail when the liquid is swirled. It is important that you don't shake the mixture, as you don't want bubbles to form. The liquid gel has to be completely clear with no gel specks before you proceed, or your DNA will run through different concentrations of agarose in your gel and cause streaky results. When the agarose is fully dissolved, let it cool to 50 or 60 degrees centigrade. This is hand hot and can be achieved by either placing in a water bath for 10 minutes or by carefully running under a tap. We cool the agarose to this temperature to prevent the perspex, which is usually used for gel trays, from being damaged. Once cooled, a dye can be added to the gel to stain the DNA for visualization. There are various dyes that can be used, but the most commonly used is ethidium bromide. Care must be taken when using and disposing ethidium bromide in both powder and liquid form, especially for pregnant women. It is a mutagen and possible carcinogen that can be breathed in and absorbed through the skin. For this reason, double gloves should be used, and when making up a working solution of the ethidium bromide, do this in a fume hood. Stocks are generally 10 mg per milliliter, and you will need 5 microliters of this stock per 100 ml of gel. This will give you a final concentration of 0.5 micrograms per ml in the gel. Ethidium bromide is a DNA intercalator, inserting itself into the spaces between the base pairs of the double helix. It yields low background and has a detection limit of 1 to 5 nanograms per band. It is very important to remember to change your gloves after handling ethidium bromide without touching anything else in the lab. This is to protect the safety of others by preventing contamination of equipment or surfaces that might be touched without gloves on. Remember that ethidium bromide will transfer to the running buffer, so this must also be disposed of safely. There are different types of gel tray. Some have ready-made ends to prevent the liquid gel pouring away while it's setting, and others do not. Autoclave tape or masking tape can easily be used to seal the ends of these trays. Tear off a piece 3 or 4 centimeters longer than the end of the tray. Remember to turn over the end of the tape for easy removal once the gel is set. Very carefully put the tape in place at both ends, making sure that there are no gaps for the gel to escape through while it's setting. Run your fingers over the edges to ensure a good seal. You will need to pour the gel on a flat surface to make sure it's the same thickness across its length. To form the wells that your DNA is going to be put into, a comb needs to be inserted before the gel is poured. The size of the comb you use will depend on the volume of the sample you have and the number of samples you want to run on the gel. Slowly pour your gel into your sealed gel tray. When pouring, it is important not to do it too quickly, as bubbles and splashes can occur. If bubbles do form, then you can chase them to the side of the gel with a pipette tip, where they won't cause any problems with the running of the gel. This is important if bubbles form around the wells. The gel sets in around 15 to 20 minutes and will change from transparent to more opaque. Remove the autoclave tape or the ends of the tray. Finally, the comb can now be removed by pulling it straight upwards. It can be quite difficult to remove the comb, but as long as the gel is completely set, the wells should still be okay.
Next, it's time to place the gel into the gel tank. The gel tank is a box with an electrode wire running across the bottom of each end. This allows the current to pass, which will separate the nucleic acid fragments. Put the gel on the platform in the gel tank in the correct orientation. Remember that nucleic acids are negatively charged, so they'll run through the gel towards the positive electrode. The connector to the positive electrode is coloured red, and the connector to the negative electrode is coloured black. Add buffer solution to the reservoirs at each end of the gel tank. Keep adding buffer until the liquid is about 2 mm above the surface of the wells in the gel. This will make the gel easier to load and ensure the appropriate current runs through the gel. You'll be able to see the wells more easily during loading if the gel tank is on a dark background. Before loading nucleic acids into the wells of a gel, you need to add coloured loading buffer to the samples. Different coloured dyes can be used, but one that's commonly used for this purpose is bromophenol blue. The loading buffer is made up of dye and a glycerol or phycol solution. The dye colour helps to visualise the loading of the samples into the wells and helps to keep track of the migrating samples. The glycerol or phycol makes the samples heavy so they sink to the bottoms of the wells. Ensure the loading buffer and the sample are mixed so the solution has a uniform colour. Arrange labelled tubes containing samples in the same order that they'll be loaded into the wells. If there are more wells than samples, avoid using the outermost wells. Samples loaded on the edge of a gel tend to run less consistently than those loaded closer to the centre. The volume of sample loaded into the wells will depend on the capacity of the well. 10 to 20 microlitres is typical. Before you pipette the sample into the well, make sure there's no air bubble at the end of the tip. If a bubble is present, gently push down on the push button until the air bubble is gone. Since the wells are rectangular, keep the pipette parallel to the row of wells when loading the samples. This orientation reduces the possibility of accidentally stabbing the walls of the wells. The tip of your pipette should be just inside or just above the well, but not touching either side or the bottom of the well. Slowly and gently load the sample into the gel, being careful not to blow air into the well once the sample's been expelled. Each time a new sample is loaded, use a new tip. When you've taken up a sample, move the tube it came from so you can keep track of where you are. Load the sample into the next well. When you've finished loading the samples, don't move the gel tank. This could knock the samples out of the well, causing sample contamination. Close the lid of the gel tank and attach the leads both at the tank and the power pack the correct way round. Set the appropriate constant voltage. This will create an electrical current through the buffer and the gel, starting the process of size separation of the nucleic acid fragments. You should be able to see bubbles rising from the negative electrode. After a few minutes, the dye, and therefore the nucleic acid fragments, can be seen to migrate from the wells into the gel, showing that the process is working. Once the fragments have separated sufficiently, it's time to visualize them. This is done with the aid of chemicals which were added to the molten gel before it was poured. Cyber green dye can be used, but the best and therefore most commonly used chemical is ethidium bromide. These chemicals stick to the nucleic acids and fluoresce under ultraviolet light. Cyber green is safer but less sensitive than ethidium bromide, which is a strong mutagen. In order to have a permanent record of the fragment separation on the gel, you need to take a photograph. This is done by placing the gel on a UV illuminator inside a box which also contains a camera. The illuminator is switched on and the gel image is displayed on a computer screen. The image can then be saved as a digital file or printed out. When heating an agarose solution, it's important that a loose top is placed on the container. If the top is too tight, there can be dangerous pressure build-up within the container, which can cause the container to break. When making an agarose gel, don't leave the gel solution unattended while it's heating in the microwave oven. A solution that's heated for too long will lose water, increasing the agarose concentration of the final gel, or even burning the gel mixture. Don't remove the comb from a gel before the gel is fully set. That will cause the walls between the wells to be destroyed and the gel will be unusable. Take care not to damage the wells when loading your samples. A damaged well can allow the sample to leak out and may cause contamination of other wells. It can also cause nucleic acid fragments to migrate unevenly. Make sure you don't load air into the wells along with your samples. Air can cause samples to be lost from the well into the buffer solution and can also cause contamination of neighbouring wells. Another mistake that can be made when loading a gel is to load the sample too quickly. This will also cause the sample to flow out of the well. Once you've loaded your samples, take care not to move the gel. Even a small movement can cause sample loss and contamination.